Hello everybody, my name is David, and I recently got braces, so please excuse my lisp. But today I'm going to be talking to you about the use of transgenic mice in cancer research. More specifically, we're going to be taking a look at the use of a green fluorescent protein in human cancer research. So where does this protein come from? It actually comes from a jellyfish called Aquia victoria, and what happens is that they take the genetic material from this jellyfish and they stick it into the mice via a retrovirus. But for this study, what they did is they took non-transgenic mice and they crossed that with a very common C57B6 mouse, which already had the gene in it. And so they crossed those two mice, a non-transgenic mouse and a transgenic mouse, and they took a look at the offspring. Today we're going to be taking a look at this paper by the Department of Surgery at the University of California in San Diego. So what they do in transgenic mouse research is that they have this mouse, and what they do is what it does is it fluoresces bright green everywhere, every single place. You'll see it right here, all throughout. Right here in part A, in box A, we have a single embryonic cell, and then on B we have a bunch of newborn mice. In part B, in box B, we have a bunch of newborn mice. And in part C, we have a full-grown mouse. Right here in box D, you can see all the various organs also fluoresce. We have the heart, the lungs, the spleen, quite a few. And then this is interesting. Part E, they have the entire digestive system. So we have the tongue, and it goes down the esophagus into the stomach. And then we have the small intestines, the colon. It's pretty cool. So here in box F, we also have the male urogenital tract. And so that means we have his testes and all his other little parts. And here we also have the female urogenital tract, which includes the vagina and the ovaries. Now here in box H, this is actually the complete circulatory system for a mouse. It has the heart right here, and you can see how it just goes, it kind of just branches out like a tree. And then we have box I right here, and that's actually the skeleton. So they took the skeleton and they put it under the blue light, the UV light, and it fluoresced. And then we have pancreas cells and as well as spleen cells. Now we're going to take a look when these mice are injected with a certain type of cancer cells. So what they did for the cancer cells is that they took them and they incubated them with retroviral cells. And these retroviral cells actually had a code for a red fluorescent protein in contrast to a green fluorescent protein. So what you're going to see here is that right here, we look at box A, you can see a breast cancer growing on this mouse. Right here in part B, in box B, we have a prostate cancer. In part C, we have a colon cancer right here. And that's where this can become really useful, because in order to remove a cancer surgically, you have to be able to see it. You have to know where it's at. You have to be able to locate it. And so after this, they took a sample of each cancer cell, and what they did was put them under a microscope. Remember that the green fluorescent proteins and the red fluorescent proteins all fluoresce under a wavelength of 470 roughly. Then we have here in box D through 8, all they're doing is zooming in from this zoomed out box D all the way into G and H. And what you can tell is that there is a cancer cell that's actually being engulfed. It's being engulfed by a green fluorescent macrophage. So that's pretty cool that they actually show up in the macrophage as well. And they've also tried a couple other things. For example, down here they talk about how they used a couple other things before. They used previously the Rosa 26 mutant line. But what they had a problem with is that it's just so easy to use a green fluorescent protein because all it requires is that blue light, in comparison to the Rosa 26, which actually required quite a bit more preparation to use. So when you think about this research, it has a little potential to change a lot of things. Right now it's only applicable to mouse research, as in hopefully someday they could use this technology to help humans. But one thing I'd like to show you is right here. This is a paper on the use of the transgenic mice in, and they are looking at, one more, there it is, human tumor angiogenesis. And what you can see right here is they use that same green and red combination. Here's a green fluorescent protein right here. You can see it for the blood vessels. And the red would be the tumor cell. The tumor cell. Okay guys, and now here we have a website that talks about the green fluorescent protein and why it actually fluoresces. And this is started by Timothy King and Paul May at the University of Bristol. It talks about a little bit about the history. The actual person who discovered it was Osamu Shimomura, and him with a team of scientists, and they used, I think I pronounced it wrong at the beginning of the video, but it's Equioria Victoria. So it's not right there. And here's actually a picture of the protein. You can see that there are 11 sheets right here of protein around it, and they actually form a barrel structure. Right here in the center, we have the chromophore right here of the protein structure. And what happens is when the photon hits that protein structure, there's a transfer of protons that go through, and when that light hits it, it'll excite it, and that's what causes that glow. But as a disclaimer, I will say I am in Chem 2, and so if anybody wants to contribute to that, probably explain it a little bit better than I can, please go ahead, leave a comment down below. One point on the ESA paper I do agree with is recommendation number 1. It is the early planning and GEO development. They should be, especially the part where it says it might include sterility, and reduced fitness. Because when you think about it, you don't really want, well, you might want a lot of, you may or may not want a lot of glow in the dark mice running around. I mean, it'd be easier to see where they are, but then again, they're easier to see where they'll be easier to locate. Ooh. And then if we look at recommendation number 3, they go and talk about preventing the release of unwanted GEOs, because often impossible after large scale field releases have occurred to control them. Yeah, and I'm pretty sure that'd be a problem with mice, because they breed like mice. Something else I would like to say is that the mice used in this cancer research are well taken care of for the most part in their facilities. The scientists that work with these animals have the utmost respect for them, and a lot of them are animal lovers, and they do appreciate the sacrifice that the mice go through. Okay guys, thank you for watching my video. I look forward to watching all of yours. Here's some of the sources I use for mine, and yes, thank you.